So my name is Mathieu Husser. I'm a PhD student at Concordia under the supervision of Dr. Piekny and Dr. Martin. I actually did my undergrad at Concordia. I come from France, but I moved for university at Concordia. I started doing some research in those labs as an undergrad, and I did my final research project with Dr. Piekny, who was interested in doing this collaboration. So I kind of jumped on that because I wanted to do some gene editing and she had that project in mind. So that's how I started in those labs. I started as a master's student and I fast-tracked into the PhD program about a year ago. And here I am. So my project mixes a little bit of both labs. Dr. Piekny most of the time studies cell division, especially the very last step where one cell physically separates into two cells, which is very important. You know, in order to make whole organisms with billions of cells, you need cell division to work. Lately, there's been a lot of gene editing that has been developed in order to study that kind of process. Without that experience, you can't really do any of that. So that's where Dr. Martin comes in because he's the genetic engineer. My research project basically involves using gene editing on human cells in order to help study the process of cell division. So a lot of what I do is take the human cell lines that we have in the lab that we culture all the time, and I introduce mutations into them. So I can delete a gene, I can make some specific point mutations that we want to study that might have an effect on cell division. And essentially, I then make a cell line that carries those mutations, and I hand that to my colleagues who will be able to study that and see, what effect it has or gather data on it.
Okay, so we're in the Genome Foundry right now with you, and you're using this specific equipment. Could you tell us about it and what's happening here?
So there's a lot of equipments that we all have access to. Specifically, I use this one a lot. It's a cell sorter. It's also called a FACS, for fluorescence activated cell sorting. Starting out with a sample of cells that are all in suspension. There's like a couple millions of cells in suspension in there. It takes that sample and flows it through different fluidic lines, and one by one it's able to detect the cells and measure their fluorescence. And so in my samples right now, I'm measuring the fluorescence of all those cells, whether they're fluorescent or not, whether they're more fluorescent than others. The second very cool thing it can do is I can tell it what cells I'm interested in, and it will isolate them for me into a tube. For example, if I want to keep only the cells that are fluorescent and the rest goes to the waste, I can tell that to that machine, and it will isolate those in the tube for me, and I can put them back into my cultures and keep them alive, essentially. So we do it purely for fundamental research. It's really just a tool. When people are interested in a specific mutation that they think might have an effect, or a specific protein that they want to be able to visualize in the cells, then gene editing is a very good tool for that. But there's other applications. Of course, you can do gene editing to treat diseases, create new therapies. It really has a bunch of applications. We mainly use it as just a tool for people to study cell division specifically. So first I'm going to resuspend the next sample a little bit because those cells are in suspension, but they tend to clump up. And this machine is very sensitive to clogging. So I'm just making sure the cells are well separated. Once that's done, I can load it into the sorter. Okay. Essentially here, what you're going to start to see is some dots popping up on all those graphs. Each dot is called an event, and it's what the detectors are detecting in there. So most of the time, one dot corresponds to one cell. So you can see it's already popping up. It goes very quickly. It looks at several thousand cells per second. What I'm interested in specifically is those graphs here, where not only am I able to see all the cells, but I can tell the fluorescence intensity that they give off. So when it measures the fluorescence on each cell, it displays it on those graphs here, and I can look at where the cells are essentially. If they're relatively dim, if they're brighter, if they have no fluorescence whatsoever. Well, something that's very important is looking at cells that have no fluorescence by themselves. So for example, here, this is what my cells would look like with nothing in them. And if I look at the sample that's running right now, you can see you still have some cells that have nothing in them, but a bunch of those cells here have more or less of that fluorescent protein in there. I can also look at different colors of fluorescence. So here I'm looking at green, here I'm looking at red at the same time. Just for us in the Piekny lab and in cell biology, what people do a lot of times is take their protein of interest or a certain pathway of multiple proteins, and in order to probe what those proteins do, they make point mutations in them that they predict would have a certain effect, and then they test that. But most of the time, how you do that is by providing those proteins exogenously. And by doing that, you're overexpressing those proteins. So in the cells, you're essentially flooding the cells with just that one protein or mutant that you want to study, and you infer whether it has an effect or not. That has a lot of problems associated with it, especially because, again, you're flooding the cells. We do gene editing, and the reason we do it right now is we make our mutations directly in the genome of those cells. So they're still expressing that protein at the same level. It's as close as possible to their physiological context. And so by doing that, when we look at the function of those proteins, because they're still at the normal level of expression in their normal environments, you essentially get more powerful data where you're more confident that the effect you're studying is actually true and not an artifact of something else. Crispr is great because you can reprogram it very, very quickly. And not only can you reprogram it, but if you want to target 10, 100 genes or even the entire genome at once, you can do it because it's very simple to target this gene or this one or this one. With previous technologies, it would take months or even years of work to target one gene or another, and so you can't do as much. Whereas CRISPR, you can do, again, tens to thousands of genes at the same time if that's what you want to do. I have an experiment where I'm looking at some human cells. Those are HeLa cells that we culture. And I want to look at their fluorescence because I have a specific protein tagged, and I want to be able to look at where it localizes in the cells. So this is an endogenous tag. So all the cells in the culture are expressing this protein with the green tag because I've made that cell line using CRISPR, and it's now expressing it stably. One thing that my cell lines are particularly interesting for is maybe having a specific protein tagged that you can visualize in the cells, and then introducing some kind of perturbation. So you can try to express another protein with a mutation, or you can try to deplete the levels of some kind of other protein, and then you can see if that has an effect on the protein that is tagged that you can follow. That's the main application. From there, you can apply it to just about anything. One challenge that we do face on a regular basis is that human cells don't like to be edited. Specifically when you want to make precise edits, so a specific point mutation that you want to make, or introducing a fluorescent tag, that kind of edits is very inefficient to introduce in human cells. And so when you have a population of cells, several millions of them, only a few of them will have the edit you want. And so part of my job is to find ways to increase the efficiency, but also to screen through all those cells to find the ones that have the edit that we want. That's not always possible. Sometimes it fails and it's impossible to introduce those mutations. Other times the efficiency is just very low, so we just need to work extra to be able to find something we want. But that is the main challenge we have.
What does a day at the lab look like for you, or a week in the lab, let's say?
Well, depending on what I plan for the day, I'll plan my experiments ahead of time, and depending on what I have to do, I'll come to the lab either very early because I need to start, or later towards 11 or 12. Usually I'll check up on whatever I did before, whether it's plates of E. coli that I have growing from the day before that I need to check up on, or my cell cultures that I need to make sure they're still alive. Then I'll usually do some molecular cloning. So I'll be in the Martin lab cloning, so manipulating pieces of DNA together for experiments that will be down the line. And towards the middle of the afternoon, I'll go to TC, to the tissue culture room, check on my cells and do anything that they need me to do. So if they're fully grown, I'll passage them to a lower density. If they need some kind of treatment or an experiment that I need to do that day, I would start that off. I try to always have something to do every day, but I can't overpack my days either. For me, I like to phrase my questions as, can we efficiently edit mammalian cells? But that is a broad question that I'm not going to answer in my PhD. If you're looking at what my tools can do, one question we're interested in is how cytokinesis, so that last part of cell division I was talking about, how that process is different between different cell types. It's something that's been studied a little bit in the last few years, and people have started to uncover some interesting things. That process would vary in terms of regulation, in terms of speed, between different cell types in a healthy organism. So developing my tools in different cell lines would allow us to study how those different cell lines undergo cell division. They all obviously do it successfully, but how it's done differently in terms of details, in terms of regulation. And that brings me to actually, we want to do the same thing in stem cells, because those cell lines are usually cancerous or they're modified to be able to culture them. Stem cells are much closer to what healthy, human or animal cells look like. So by doing the same things in stem cells, and then we can differentiate them into different cell types, we can study those cell types and how cytokinesis, again, is different between those cell types, which would reflect the actual cells in a human body. We've sent some cell lines to collaborators in other labs in Montreal, one in Germany as well. We have other ongoing collaborations with different labs at Concordia. Just us using these equipments and having Nick and Smita, who are here all the time, kind of helping us develop protocols on those machines is really a really nice aspect to my work, yeah. It's great to have other people who, ask me for those tools, even, halfway across the world, because they're studying the same thing and they think that tool can be valuable to their research. I love that.